Good day to everyone joining us, and welcome to today's X Talks webinar. Today's talk is entitled Recombinant Protein Expression in Pharmaceutical Industry Success in CHO. My name is Sonia Hunt, and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speaker throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank GenScript, who developed the content for this presentation. GenScript is the leading contract research organization in the world, providing gene, peptide, protein, CRISPR, and antibody services. Since its foundation in 2002, GenScript has grown exponentially through partnerships with scientists conducting fundamental life science research, translational biomedical research, and early-stage pharmaceutical development. The company is recognized as having built a best-in-class capacity and capability for biological research services, encompassing gene synthesis, peptide synthesis, custom antibody and protein engineering, and in vitro and in vivo pharmacology, all with the goal to make research easy. Now it's my pleasure to introduce you to your speaker for today's webinar, and she is Ishita Barman, Senior Field Application Scientist with GenScript USA, Inc. Ishita has over 13 years of extensive hands-on experience in a broad range of molecular biology, expression, and protein engineering skills. She has worked with therapeutic antibodies in pharmaceutical companies like Pfizer and BMS, where she actively participated in early research and drug development. She is currently a Senior Field Application Scientist with GenScript's Life Sciences Group, focusing on their protein and antibody portfolio. Ishita holds a master's degree in molecular biology from SJSU, a MS in microbiology, and a BS in biological sciences from MSU, India. Now, it's my pleasure to pass over the mic and the controls over to Ishita. So Ishita, when you're ready, you may begin.
Thank you so much, Sonia. So I will be talking about recombinant protein expression in the pharmaceutical industry, success in CHO. I am Ishita Varman, and before I jump on to the actual topic, just a little bit of introduction about myself. As mentioned by Sonia before, I do have a background in microbiology and biological sciences. I was born and raised in India and then moved to San Francisco, where I have quite a bit of research experience. Currently, I am a senior field application scientist working on the protein and antibody portfolio for GenScript. However, in the past, I was 100% bench scientist and worked at Pfizer and BMS, also in the protein engineering department, doing a lot of protein expression, molecular biology, purification, cell-based assays, and so on. I did work in a small startup as well, doing therapeutic discovery, where my focus was on yeast display platform, protein expression, and purification. So this is going to be the outline of my talk. I will start off with a little bit of company overview, introducing GenScript, and briefly go over what we do. I will then transition into the basics of protein expression, and finally focus on the CHO-Expression platform. I will close today's talk with sharing some CHO case studies. So a little bit about our company. This is the history and milestone of the GenScript group. We started out in New Jersey in 2002. They were founded and we launched the first custom gene synthesis platform. Now this is a very comprehensive overview, but I have highlighted milestones here in green, which indicates new subsidiaries that we have launched. For example, Bestzyme, which deals with a lot of industrial synthetic biology products. Legend Biotech, which specializes in cell therapy. and more recently GenScript ProBio, which specializes in biologics CDMO. What I have also done in blue is indicating landmark achievements for us where we have been able to successfully collaborate with other companies, for example, in this instance, Janssen, and have put molecules into the clinic or IND. For example, BCMA mentioned here, where we have entered into collaboration with Janssen. The program also received orphan drug designation by FDA. And in 2018, BCMA program received IND clearance in US and China. We have also been able to focus quite a bit on our CAR-T program. Clinical data showed 100% remission rate. So all that I wanted to show here is not only are we a CRO company, but we have also been actively participating in putting molecules in the clinic. This is continuing along the same lines of the company overview. More importantly, we are a platform company. We have a culture of innovation, and we have also a proven track record of incubating several internal unicorns. And because we have these small mini branches, we work together and build up a portfolio that leverages the strength of each individual business unit. We also have a strong IP position across all business lines, and we have over 140 granted patents and over 500 patent applications across all these 4 platforms. We are a global business. We cater to 100,000 plus customers in over 100 countries, and our global sales, manufacturing, and R&D presence is worldwide, including US, Europe, and Asia Pacific. We are a global company and employ more than 4,600 employees, and a lot of them have a higher degree of education. So these are the 4 core business platforms. First is life sciences products and services, and part of what I'm going to talk about today falls under that category. Followed by the biologic CDMO, which is a collaborative effort where clients come to us when they're ready to go to the clinic, and that is where ProBio steps in and helps with developing and taking things through up to filing INDs and up to clinical trials. We also have our third subsidiary called Bestzyme, which focuses mostly on industrial synthetic biological products. And last but not the least, Legend Biotech, which is focused totally on cell therapy. These are some of the branches and areas where GenScript life sciences products and services are widely used, starting with gene synthesis, peptide synthesis, protein production, which is something that I'm going to touch upon very briefly today, antibody development. I will cover that in the following slides as well. We also provide research reagents and equipment, catalog products, as well as oligonucleotide synthesis. So when we have a pipeline which is as diverse, we are able to help in different aspects of life science research and applications, starting with just basic research, protein and antibody engineering, biopharmaceutical, gene and cell therapy, synthetic biology, as well as diagnostics. So that gives you a little bit of idea about the company as such. So now I will move on to the basics of protein expression. Before I go on to talk a little bit more about the CHO cell, I just wanted to revisit and share some insights on the industrial applications of protein. So what has protein empowered? There is applications of protein are all along in the biotechnology industry, food, agriculture, therapeutics, functional genomics, drugs, antibiotics, and incorporates one or multiple of these modalities. We have transmembrane proteins, enzymes like kinases, co-express proteins, cytokines, vaccines, growth factors, viral envelope proteins, nuclear hormone receptors, antibodies. So the application field is extremely diverse. So there are broadly 4 different expression systems that we use on a routine basis in GenScript: bacteria, yeast, insect, and mammalian. So for our bacterial work, we pretty much focus mostly on E. coli, and we are also adding Bacillus subtilis. And the reason for that is we have to deal with less of the endotoxin problems when we do Bacillus. There is also yeast. Insect, we have a diverse array of cells, for example, SF9, Hi5, and mammalian efforts are mostly focused towards HEK293 and CHO cells. This slide explains the different expression systems and goes into detail about a little bit of what each system can accomplish. So E. coli is very well established. The genetics are simple. Scale up is very easy. It is very speedy in the sense that proteins can be expressed overnight, and also very, very cost effective. For insect platform, there are post-translational modifications. However, the proteins are soluble and usually used when they're really complex and also large proteins. The yeast platform will involve proteins that typically have post-translational modifications, soluble proteins, and higher expressors. The mammalian platform, including CHO and HEK293 cells, will include a lot of cells that will include modalities that will have post-translational modifications, soluble proteins, and are quite expensive. We have expressed a wide range of targets using all these expression systems, starting with antibodies, bispecifics, kinases, cell surface receptors, cytokines, DNA binding proteins, nuclear receptors, proteins from natural resources. The targets are limitless. Which brings us to our first poll question, and I will hand it over to Sonia.
Oh, thank you so much, Ishita. Yes, I'll conduct our first poll question. So audience members, you'll see it appear on your screen right now. And it's very simple. All you have to do is select one of the below. I'll give you about 40 seconds to complete this simple poll question. And the question that we have for you there is, what expression systems do you use in your lab? E. coli, insect, mammalian? Please go ahead and choose. Now, this is done in real time, so your participation is strongly encouraged and very much appreciated. Once I close the polls, I'll share the results with everyone, and then we'll go right back to Ishita. Okay, that looks like it's the majority of our audience members, so thank you very much, everyone. I'll give a few more seconds. It looks like there's a couple more people still conducting this poll. Okay, that looks like it's everyone. All right, thank you, everyone, for participating. Let's take a look at the results. Okay, so the results are here. We have 42% of our audience members saying E. coli, and then we have 3% saying insect, and 56% are saying mammalian. So thank you, everyone, for participating in poll question number 1. Now back to Ishita.
Thank you, Sonia. Thank you, audience, for participating in the poll, and I will go back to my presentation. So this is a typical workflow of protein expression. Every protein expression, as some of you are aware, starts with gene synthesis. So once we have a target protein, we get the sequence, and the sequence can be either amino acid sequence or it can be a DNA sequence. This is followed by generation of an expression construct, because once the protein is expression construct, once the protein is expressed, it'll have to be, it is then purified, and then there are quality control procedures in place, which is typically running a gel, making sure that the protein looks okay, as well as doing some additional QC features. This is a week-by-week split between the 3 systems, and I did not include yeast here because that is not one of our more popular systems, as you have already seen before from the poll results. So comparing the different systems, as you can see, the mammalian system is the fastest, where gene synthesis as well as expression, purification, and QC can be finished within a span of 2 weeks. So it's total 10 business days from gene synthesis to delivery. Insect cell takes the longest because there is a step for generating baculovirus and expression. And then E. coli is, will rank the second in terms of speed. These are the characteristics of the 3 main expression systems. A little bit of more in-depth analysis between the analysis and comparison between the 3 systems. We have the categories, timeline, which is the fastest in E. coli, slowest in insect, like we just saw. Mammalian is transient. So here, if you're looking at 2 days, insect will be weeks, and mammalian will be anywhere between 1 to 2 weeks, as we saw in the previous slide. The price point here with E. coli is low. It is very, very cost effective. With mammalian, it will be high because of the media that is involved, the feed, and several other plasmids and cells as well. Expression level will vary. Here it's just a comprehensive overview, but protein to protein expression levels will be varied. However, as a general rule, E. coli is generally high, insect is low to high, and mammalian transient will be high. Protein localization, what are the usual characteristics when clients come to us? For E. coli, it's usually intracellular, insect is extracellular, and mammalian is extracellular. An example would be secreted proteins, antibodies. For insects, it's extracellular and secretory proteins. Some proteins are difficult to express, such as membrane proteins, kinases, proteases. In terms of E. coli, it would be any intracellular protein, tool enzyme, antigen that can be expressed really quickly. Post-translational modification, very important depending upon the downstream applications. For E. coli, there are none. So if somebody is looking to just use the protein in downstream assays and are not worried about a lot of post-translational modification, this is the system to use. Keep in mind, this is the fastest and the price point is also very low. For insects, it's anywhere in the median. It is simple glycosylation. However, mammalian cells come with comprehensive glycosylation, which we also refer to as post-translational modification. Technical difficulty starts with low in E. coli and then becomes high as we move to insect and mammalian cells. This pipeline shows drug development towards commercialization, and it's an illustration from early stage discovery to commercialization of proteins and antibody therapeutics. I have shown here how the HEC and the CHO cell usage is usually in the industry. As you can see, anything that is gene to target here highlighted in these two blocks is part of early discovery. So typically in early discovery, you're still trying to figure out what the genes are, what antibodies you're looking at, you're selecting candidates, you're doing target identification. And if you think of it as a big funnel, this is where the funnel is at the widest part. And then you're trying to narrow down the funnel and pick the top 5 or 10 antibodies, which takes us directly to the lead, to identifying leads. So here we have lead identification and validation, lead optimization. And as you can see, initially when you need only microgram quantities of protein to narrow down the discovery process, lead identification, validation, optimization will require a little bit more, anywhere between 500 milligrams to 1 gram. And at this point, you also will not have a high number of candidates. You might possibly have top 5 or top 10 leads. And this is where people would come in and do in-depth biophysical characterization, cell-based assays, biological assays, and so on. Finally moves to candidate selection, starting with preclinical and GLP toxicity studies, some of which are also done in animals. And that is why you'll see the amount of protein required increases from 1 gram to 10 grams. Patient response, going into clinical trials, phase 1, 2, 3, as well as commercialization. Typically, transient expression is used for antibodies here, leading up to the target lead optimization, and thereafter, it slowly transitions into stable CHO. And the reason for that is you need gram scale quantities, as you can see here, and it is much more economical and time efficient to move on to a stable platform. So traditionally, HECs have been the workhorse in early discovery, moving on to CHO. But when you're moving from one system to another, there are 2 different biological systems, and sometimes post-translational modifications can be different, and proteins can behave differently in 2 different cell lines. So the reason, part of the reason we wanted to introduce our new CHO platform is because it gives you a continuous workflow starting from early discovery all the way up to commercialization. And I'll get to those slides in a little bit. So early identification of negative characteristics increases the probability of successful transition from phase 1 to final approval. Also, it'll be very, very beneficial if we can expect some idea of the protein characteristics, biological activity, stability, purity at an affordable price in advance. So if it is a candidate that is not well behaved and we come to find out during one of these early stages, it will be very, very beneficial so that we can move on to some backup candidates. And a lot of times that is hampered because most of the studies are done in HEKs versus CHO cell lines, although things are changing now. So a good case in point is antibody therapies against cancer. So the therapeutics can come in different flavors and different flavors, where we would have an example would be enhancing active effector functions by way of ADCC. We can also do directly arming the antibody using radionuclides, immunocytokine. A lot of cytokine engineering is coming up, protein toxins, small molecules. Indirect arming using scFvs and immunoliposomes, radionuclide, bispecific antibodies using the killer cell approach, and as well as pre-targeting. So the antibody is made such that it is not available until it hits the target. So it's usually in the pro-drug form, and then there is a cleavable reaction happening so that then the drug gets activated. On the right-hand side is a conventional IgG, and things are changing drastically in the last few years, and people are moving on from conventional IgGs to new modalities. Here I'm showing you a bispecific T-cell engager, what we call BITE. You have two different scFvs targeting two different receptors. There are DARTs. These are just examples. There are heterodimeric IgG-like bispecifics as well as 2-in-1 bispecifics. Some of the therapeutic antibodies in the market, are here on the right-hand side: Rituxan, Humira, Prolia. However, the global biologic forecast is looking at around $394 billion by the year 2024. The reason I'm bringing all this up is the market is growing, rapidly growing. And in that, majority of the monoclonal antibody therapeutics that were approved between 2014 and 2018, they were all done using CHO cells. So now I'll move on to the CHO expression platform. Before I start talking a little bit about what we have been able to achieve, I just want to give you a little bit of information, background information on the CHO cells. So what are CHO cells? Some of you may already know this, but for those of you who are not very familiar, CHO stands for Chinese hamster ovary. They are an epithelial cell line derived from the ovary of Chinese hamster, as the name suggests. Under the microscope, this is how it looks like. It is used in biological and medical research and is the most commonly used mammalian host for industrial production of recombinant therapeutic antibodies. So this is the hamster. This is how it looks like under the microscope. And this is showing monoclonal antibody therapeutics that have been approved using CHO cells. As you can see, a major chunk of the approvals have come from CHO-derived material. So this brings us to the evolution of CHO cells. Now, as you can see, they were developed. CHO cell populations were handled by a number of different labs, passed on to many different researchers in this field, under many names that are not very popular anymore. For example, we have CHO-Ori, and then we have CHO-Toronto. These are some examples. Some of the following names are still in use today, and I'll go over that in the next slide. For example, K1 and DXB11. But what I'm trying to show here in this slide is, while there is some literature available that provides the locations and names, it is very difficult to track precise progeny of these different cell lines and their number of cultivations or their number of divisions and progeny. So this is again so taking the information from the previous slide, this is just highlighting the main features of some of the CHO cells that are currently in use. Starting with CHO, the original CHO line, which was established in the lab of Theodore Park in 1956. However, in 1968, it was cloned in the same lab. This particular K1 variety, K1 isotype, is missing a chromosome. And it's missing a chromosome which carries the gene necessary for glycine biosynthesis. Followed by CHO-S. S stands for suspension. This is a cell line, a CHO cell line that has been adapted for growing in liquid culture. And what it does, it enables scale up and growth in large scale reactors. This was followed in 1980 by a DHFR diffusion system. There is a deletion of this particular enzyme on the locus, and what that does is it gives us opportunity for downstream screening purposes. So in terms of the possibility of selection of cells carrying the gene of interest, and it's co-transfected with another DHFR gene and gene of interest. CHO-GS was introduced, which allows MTX-induced gene amplification, followed by, in 1983, CHO-DG44, which has a full deletion of both DHFR loci, and several other improvements along the way, ultimately bringing us to the 2000s, where already, they have been in the market for quite some time, and 100+ new recombinant protein therapeutics have been used and approved. Some of the advantages of CHO-expression system over HEX. The productivity here, I think I'll highlight the most important point, which is the productivity. Here it's medium, and here it is high. Growth in serum-free medium is possible here. GMP production is possible, but not common. And for CHOs, they have been used for greater than 85%. The discovery, as we saw, is 1956. Biosafety, it is resistant to a number of viruses. Post-translational modifications are here in both, so I don't think that is a major advantage, but however, it confers more human-like glycosylation here in terms of CHOs. So the CHO platform can be used as transient as well as stable CHOs, and this is a comparison of the workflow and timeline for transient versus stable CHO. What we have been able to do here at GenScript is we have a transient system. Starting with gene synthesis, we can do a high efficiency transfection in 5 to 10 days, and then followed by protein production. The stable expression and comparatively takes more than 3 months, because here it starts with transfection followed by expression, selection, and subculturing. And the way the stable expression works is you have to continuously select out cells for single growth, single clones, and that is what takes time and is also very labor intensive. So to compare between the two, we have 5 to 7 days for transient cells. Stable cells take much longer, greater than 3 months. It is suitable for one-time expression, and if there's a long-term project, like I mentioned, when companies are going into clinical trials, they need they do not want batch-to-batch variability. They need something that can be consistently used. They make one big batch so that we can take care of the other variability down the line. So once we have a long-term project, stable cells is the way to go. Application mostly in vitro testing, and stable is used mostly in the preclinical stages. As I just mentioned, there is batch-to-batch or may be batch-to-batch variations. However, with stable pool, it is consistent. So some of the common challenges in protein expression and how can CHO cells help? So, as researchers, some of the puzzles and problems, challenges that you might be facing is which system should you choose? Should it be HEKs? Should it be 293s? You're currently screening an antibody-based drug candidate. What is the fastest way? You need more and more antibodies. There are different antibodies. How can you get to your research faster? The target protein expresses has a very low yield in HEK 293 cells or other expression system. So how can you counter that challenge? If everything else around there, other options are too expensive, your budget is limited, how can that be helped? If sometimes it is taking 4 months to get your antibody at hand, that is a big no-no. That results in a lot of time spent upfront. So are there any solutions? At this point, before we look at the solutions, I would like to hand it over to Sonia for our next poll question. Thank you, Sonia.
Thank you. All right, here's our poll question number 2, everyone. I'm launching it now. And just like before, I'll give you 45 seconds to complete this simple poll question. What does your work mostly involve? Recombinant proteins or recombinant antibodies? And once I close the polls, I'll be able to share the results with everyone, and then we'll get right back to the presentation with Ashita. Oh, that's great. Looks like that's majority. I'm just going to keep it open for a few more seconds for those of you who are still deciding. If you can go ahead and just click on one of the below. Perfect. That looks like it's mostly everyone. Thank you very much. I'm going to close the polls now. Okay, so the results are here. We have in our audience 70% are saying recombinant proteins, while 30% are saying recombinant antibodies. So thank you everyone for participating in poll question number 2. Now back to Ashita.
Thank you, Sonia. So we left off with our previous question asking how can we help researchers solve their problems of protein expression. And we just wanted to introduce this slide where we are coming up with an optimal solution. We have an advanced TurboCHOT platform that we just launched. And to address one of the most common concerns, the target protein has a very low yield in HEK293 cells or other expression systems. What can we do? We are looking at an alternative approach for difficult and low yield protein antibodies. We have noticed yield improvements of up to 100-fold in some cases, quick turnaround time from sequence to protein in as little as 6 weeks, and we can also go up to gram level expression amounts. So this advanced TurboCHOT platform is a cutting-edge system that enables generation of a functional recombinant antibodies or proteins at much higher yield in comparison to regular transient expression systems. The other question and challenge that researchers often face is high throughput gene-to-antibody platform. How can we, in terms of antibody drug-based screening, what is the fastest way? So our current platform hopefully will be able to solve that problem as well, because we have a large capacity and can run almost 10,000 targets per month. Our options are also flexible. It is applicable to various targets, including Fabs, SCFVs, VHH, and other fusion proteins with FCs. It is also cost effective. It is standard. There are different options and a 24-well plate option available as well. It is also fully automated, which means that there are going to be fewer human touch points and very little chance of cross-contamination. It takes almost up to 2 weeks to deliver antibodies starting from sequence scratch. However, we can also do antibody drug screening, engineering, ligand competition, binding assays, affinity measurements, epitope binning, screening assays. And I did mention antibodies here, but I also want to remind you that this is also a very good system that can be used if you're looking to do some enzyme engineering. You want to test out different variants of enzymes in a small amount of time, and you need a small quantity of protein to do your downstream assays. This is also a very good platform to use. So we are using engineered CHO-K1 cells for optimal productivity. We took the CHO-K1 cells and they were engineered in-house for giving higher titers. And this is revisiting the previous slide. But what I'm trying to show here is we are trying to use that from all the way from gene to target up until commercialization. And the thing is, I have highlighted the different scales of the CHO cells that we are using here. We start with CHO-HT. HT stands for high throughput, followed by CHO-Express. CHO-HP is high performance or a stable pool, followed by ProBio, which is our subsidiary or our sister concern that takes care of all the commercialization. So there is minimal risk in the sense that you're expecting similar biophysical and biological characteristics and post-translational modifications. At the same time, one very important thing to keep in mind is you're starting with the same CHO-K1 cell. You're starting with that parental cell line here all the way up to commercialization. You're using it in early research and discovery, preclinical and clinical. So there are no surprises in the end in terms of the protein quality and yield. So this is some more details about the new CHO platform. There are several scales available, and we have designed this keeping in mind that not all researchers might need a large quantity of protein to start with. This is the timeline in weeks. So CHO-HT from start to finish, and start is gene synthesis up to protein purification and QC is 2 weeks. And with CHO-Express, the volumes are also, the amount of protein is also slightly higher. It takes an extra week. So we are looking at anywhere between up to 5 milligrams for CHO-HT, looking at around 500 milligrams for CHO-Express, and for CHO-HP is going to be gram scale. We also have a CHO stable cell pool. So sometimes if I recommend this to scientists who are in a hurry to get a little bit of material, so we can get them started with either CHO-Express or CHO-HP. But if you know that you are going to come back and use that protein or that antibody later in bigger quantities, an option would be to also start the CHO stable pool side by side. So we can give you a little bit upfront. We can deliver a little bit of your protein for your immediate needs, and then in the back burner, this is also happening. So in 3 months' time, you will also have a CHO stable pool and not have to worry about replenishing every time. So now that we've heard so much about the TurboCHO, let's see how the yields are comparable. So we have taken, this is showing only antibodies, human IgGs 1, 4, mouse IgG2A, rabbit. So different isotypes, different subclasses, and different species. And the yields have been quite good. So on an average, we are looking at around 250 mg per liter. What I have on the y-axis is the yield, and then the x-axis are the different species and subclasses. For the TurboCHO-Express, which is one scale higher in terms of expression, we also notice that the titers are pretty much around the ballpark of 250 mg per liter. This is the CHO-HP, which is the high performance. We tested 7 antibodies, human and mouse combined, and we have been able to come up to an average of 2 grams per liter. This is showing the black bars are the CHO-HP system. This is a previous system that we were using at GenScript. So in some cases, we have had extremely significant improvement in yields. So this is this slide is giving you another overview where it categorizes what are the scales and what are the QC options. For the high throughput, anywhere from 100 micrograms to 5 milligrams protein is can be achievable. However, keep in mind this is the data pertaining only to well-behaved antibodies. We do this also for proteins. CHO-Express anywhere under 500 milligrams, and for CHO-HP anywhere from 500 milligrams to gram scale. So the QC features, this is very important. So what we are offering here is for the high throughput, since at that stage you're just screening candidates, we thought it may not be economical to offer endotoxin or mass spec. However, those can always be added on. We offer basic A2AT and an SDS page to check the purity. For CHO-Express, we do offer a more comprehensive QC in the sense that we offer analytical HPLC and endotoxin testing. Analytical HPLC will tell you the integrity of the protein. So you have 500 milligrams, but you don't want majority of the 500 milligrams to be oligomerized, aggregated. So that's what we can check with analytical SCC. We also have endotoxin data provided, and if you tell us what endotoxin levels you're looking for downstream assays, for example, if you're doing cell-based assays or using this for injecting animals, we would like to keep the endotoxin low, and that's where this comes in really handy. However, in addition to this, there are also other features that can be added on. For example, you can add on analytical SEC to the high throughput platform. You can also add on LC mass spec to the CHO-Express platform. And additionally, we can also do affinity testing, cross-reactivity, epitope mapping, stability test by freeze-thaw, and so on. In terms of purity, these are some of the values that we have available, and we are consistently meeting these requirements when we make the proteins in-house. The timeline is important. We have 2 weeks high throughput here, and that increases by 1 week when we are doing the CHO-Express platform, because it also the culture volume is a little higher, and then we also have to scale up the plasmids. For the CHO-HP, which is a stable pool, it is around 6 to 7 weeks. That brings us to the third poll question, and I'll hand it over to Sonia. Thank you.
All right, yes, it's our final poll question, everyone. So I'm going to launch it right now, and you know what to do. So please pick from the below, what kind of proteins do you work with? You have options of secreted proteins, intracellular proteins, or transmembrane proteins. So please go ahead and cast your vote, everyone. I'll give you 30 seconds to complete the simple poll question, and then we'll go right back to Ashita. Okay, that looks like it's everyone. I'm now going to close the polls, and let's take a look at the results. Okay, so our audience out there is saying we have 57% are saying secreted proteins, Then another 27% are saying intracellular proteins. And then we have 16% of our audience members are saying transmembrane proteins. So thank you everyone for participating in our final poll question. Now we're going to send it right back to Ishita.
Thank you, Sonia. So now we are at the last leg of my presentation, which is the most important part according to me. I gave you so much information about CHO cells, protein expression. So how does this platform work in our hands? So I will be wrapping up with some CHO case studies. So what I'm going to be showing here are mostly antibodies, and I know there are also there are also audience members who are not doing antibodies. And I also have information regarding that, and I'll get to that after I finish up with this portion. So please hang on there, hang in there. So we have full-length IgGs that I'm going to show you in different species and subclasses. And I will also wanted to show you some next generation antibody modalities. So some of the case studies are here from that I'm showing you here, typically with an SDS page, as well as SEC HPLC, showing you the integrity of the protein, looking at rabbit antibody, human IgG1 and human IgG4. These are all full-length normal antibodies, and you can see from the SEC profiles, they're 99% pure and monomeric. You can also see from the gel pictures that the preps are pretty clean. So this is full-length IgGs. Now, how are we doing with the next generation antibodies, and why are they important? So we know that the human design next generation antibodies are becoming breakthrough therapeutics of today. And this is, I have listed some new modalities here. For example, I will bring up the first FDA approval of the VHH, which is Cablivi. I have mentioned here by Sanofi. Other modalities include SCFVs and CAR-Ts, bispecific IgGs, BiTEs, not new anymore, it has already been 8 years. So let's see how we have been doing, performing with these new modalities. Some additional ones that we have produced, Fabs, full-length antibodies we already looked at, single-domain antibodies, SCFVs, BiTEs, bispecific antibodies in different forms. So what I am showing here is a particular single domain antibody that we expressed using a prior existing platform and the new platform that I just talked about, the TurboCHOT. And you can see that the yield has increased by quite a bit. These are the two different purification proteins from the two different purified sources, and they look pretty similar. All that has changed is the yield has become, it's a lot higher yield. This is another bispecific, reduced and non-reduced gel. The preps are very clean. However, one important thing to keep in mind as always is the purity, which is 99% monomeric. This was a knob and whole backbone, in case you're wondering, based off of the cross-map scaffold. In terms of nanobodies, this is an example. It is right around 15 kilodaltons, and the yield was pretty high, around 510 mg per liter. We are looking at around 98% monomeric protein. So that was a very well expressed nanobody, but as you know, antibodies, their proteins are different from one to another. This is another case of nanobody. Although the preps are clean, we do notice that the average yield was slightly lower. It was 27 mg per liter. However, purity by SDS is around 95%. When we do have poor expressors, all that we do is we can suggest clients to go the CHO-HP route and get enough protein to satisfy your research needs. Some of the antibody fragments that we have produced, SCFVs, they are all around 25 kilodaltons, and the yield has been 94 mg per liter. Again, wanted to draw your attention here to the purity, which is 99%. Same here for the VHH heavy chain construct, yields looking at around 135 mg per liter, and this is the integrity. So when we do all this work, these are some of the purification methods that we have, that we use. Starting with size exclusion chromatography, we do hydrophobic interaction chromatography, ion exchange, as well as affinity chromatography. If we have the FC, we typically resort to affinity chromatography, and then if there are aggregates, we would do a final polishing step that includes SEC. Sometimes if clients do want endotoxin control, we do have operational controls where we keep everything clean and do multiple washes between runs so that there is no cross-contamination and endotoxins are removed. We also do chromatographic removal of endotoxin using ion exchange. If there are proteins that have a tag for expression and purification and then the tag needs to be removed, we do that as well. Of course, keeping in mind that we would lose a little bit of protein, so we factor that in when we do our initial calculations in terms of protein, how much protein and what cell volume to use. We do provide lyophilization as well as concentration. Some of the QC specifications, we do SDS-PH for purity. We do provide SEC HPLC for homogeneity. Are there aggregates? Is it monomeric? To look at the quality of the protein that has been delivered. We also do LCMS, endotoxin, and freeze-thaw. However, we are also researching and trying to build up facilities to address thermostability using DSC, SECMOLs, SPR, and other downstream functional cellular assays. In terms of QC platform, just a little bit more information. We do have LCMS, we do have HPLC, we provide concentration, SDS-PH, Western blot, and endotoxin, something along the same lines as what I just showed in my previous slide. So in order to do all this work, just wanted to give you a glimpse of our facilities. We are very, very well equipped to do the runs that we mentioned. We do have big batch Cooner shakers, wave bioreactors, a Sartorius fermenter, and Agilent LCMS instruments. We have several Actopures for protein purification. So, and all this instrumentation allows us to reach the high levels of productivity that we are showing here. So to wrap up, what I just talked about here basically is a proprietary CHO system. And for the smaller scale, we do deliver in as fast as 2 weeks. It's also a automated high throughput platform with comprehensive downstream services. There are fewer touch points and therefore fewer chances of cross-contamination. We have successfully delivered almost greater than 50,000 proteins, antibodies included, and we also have extensive experience in not only different antibody formats, but also a lot of other recombinant proteins. We can go from milligram scale to kilogram scale, gram or kilogram scale, and therefore offer a complete solution for antibody drug discovery, because we can also partner with our subsidiary group who takes care of the commercialization. We have batch-to-batch consistency and delivery in as fast as 2 weeks. That brings us to the end of my talk. And I do remember, before I end, I do want to show you some non-antibody protein expression using the CHO-Express. These are some fusion proteins and some recombinant proteins, but what I am showing here is the CHO-Express is in green and the competitor is in orange. I wanted to show that even with non-antibody proteins, our titers have gone up to as high as 450 mg per liter. There are some outliers, some places where we have not been able to help as well as we would have liked to, but these are our expression levels in the CHO-Express. The next slide has the CHO-HP, which is stable pool, just as a reminder. And there as well, we have outperformed our competitor. For this particular instance, if you zoom in, we have reached up to 1.2, 1200 mg per liter. And then this one is around 2.5 grams per liter yield. So something to keep in mind for all of you in the polls who said that you're also interested in non-antibody proteins. Now that brings us to the end of my talk, and I will hand it over to Sonia. Before I do want to thank all of you for your time today and hope this was useful. I look forward to hearing from you. Thank you very much.
Oh, well, thank you very much, Ishita, for that very detailed presentation. I'm sure everyone enjoyed it. And with that, we're going to put on our webcams to begin the Q&A portion of the webinar. So I invite Ishita to do the same. Hi, everyone. And now remember, you can send in your questions by using that questions chat box that's located on the right-hand side of your screen. And we'll try to attend to your questions during the time that we have together. Now, I've received a bunch of questions while you were speaking, Ishita, so let me just get them all lined up here so that we can get to it because we have a limited time. Okay, so let's go to our first question. Now, this audience member is asking, how long does it take to make recombinant protein with your advanced platform? It's a good one.
Thank you. So the time would depend upon how big your protein is. However, the smallest and the fastest that we can do is starting from 2 weeks. And that starts from gene synthesis all the way to purification. But everything is customizable. If your protein is too big, the gene synthesis might take a little longer. So that would delay it. So I would say depending upon the length of your genes as well as how much amount of protein you need. Those are the two factors. Thank you.
Thank you. I'm busy here looking at all the questions that are hurriedly coming in. All right. Thank you for answering that one. Let's go to the second one. Okay, so this person is asking, how much does it cost for CHO-HT for recombinant protein production?
Our package is very competitive. I don't remember over the top of my head, but if you do reach out to me separately, I can direct you to our salesperson. I'm a field application scientist, and I might not be the right person to give you the information.
Thank you, Ashita. All right, let's go to the next one. This question is, could you please talk a little more about the monoclonal selection step and the bioprocess scaling step?
So monoclonal selection step, if you're talking about which phase, I'm not sure. Monoclonal selection. Bioprocess, I do understand what you're saying. So for the monoclonal selection step, in terms of the stable cells, if that is what you're asking, we do a limited dilution and try to adjust. Once we do our transfections, we let the cells rest, and then we start doing our screenings. And in that screening, what we try to do is we try to plate the cells such that there is one single cell clone per well. It's called limited dilution. And from there on, we would have to monitor the cells for their productivity, which we do using an ELISA. So we have to, not only do we want single cells, but we also want good producers. And from there, we scale up the cells and such that we change media and successively change takers and containers, so that we start with 25 mLs, go on to 100 mLs, and up to a liter. And once we go there, we freeze down the cells and store them, so that next time we need that, we just pour out a small vial, and go from there.
Okay, thank you. All right, we still have time. The same person is asking, also, does your company have a proposed protocol for protein expression in advanced TurboCHO system?
Yes, we do. Please reach out to us, and then I can help you with that and direct you to the right people.
Okay, all right, thank you. All right, let's go to the next question here. This audience member is asking how to calculate expression level and purification level.
How to calculate?
Mm-hmm.
I'm not sure there is a way to calculate if you're trying to gauge how your protein would express. Unfortunately, there is no way to do it yet. But if you do typically what we suggest is, if clients are coming to us for the first time with a protein they have never expressed, we do a small pilot scale and we see how the protein is expressing, and then taking that as our starting point, we can come up with a particular volume that will give you the amount you want.
You've got all the answers, don't you? Okay, Ashita, thank you very much for that. Let's go to the next one here. What is the difference between your CHO and your competitors? What is the difference between that? This audience member is asking.
That's a very good question. So for our CHO cells, yeah, thank you for that question. So our CHO cells have been engineered in-house at GenScript, and we have made certain modifications to the cell line. This is a CHO-K1 cell line. And at this point, we are not, it's proprietary, so I cannot discuss exactly what changes we made. But we have engineered the cell line for better production in-house. And the other thing that we have also used is we use our proprietary plasmids. So it's a combination together. With the plasmids, we have used better enhancers. And we have also optimized the growth conditions using our own proprietary feed, media, and so on. So the whole package comes together very nicely and results in the higher yield.
Okay, thank you. All right. I think we have time for one more question. I'm going to squeeze this one in. This audience member is asking, what are the protein QC metrics when using the CHO expression platform?
Yes, some of the protein QC metrics that we use are typically we would do a A280 that will tell you how much protein there is. We do an SDA spade gel, but there are also other additional add-ons. For example, you can add on analytical SEC to see the integrity of the protein. You can also add on LCMS, mass spec. You add on endotoxin. And there are also a lot of downstream affinity measurements that we can add on. So it's very, very customizable. And if you have specific requirements that you don't see here or you didn't see in my slides, please let me know and we can work. We can see if we can accommodate your request.
Okay. All right. I'm squeezing one more in. I think we have time. Have you expressed non-antibody proteins in the CHO platform, and what are the expression levels?
Yes, so we have expressed non-antibody CHO proteins, and the last two slides that I was able to squeeze in, they are typically around 2.5 grams per liter for the CHO-HP, which is the stable pool. And for the transient, it is anywhere around 400 mg per liter. But then again, protein to protein is different. So I typically always recommend clients to do an initial evaluation with us.
Okay, well, thank you very much, Ashita, for that. Well, thank you very much for those questions. We have reached the end of the Q&A portion of the webinar. If we could not attend to your questions, the team at GenScript will follow up with you after this presentation. And please, if you have any further questions, please reach out to, use this email address here, protein@genscript.com. Thank you everyone for participating in today's webinar. You will be receiving a follow-up email from XTalks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now give me a few seconds. I'm going to send you a link in your chat box. You'll be able to view the recording of this event with that link and also share the link with your colleagues once they register for the recording as well. So I encourage you to do that and get even more out of today's presentation by downloading the supporting materials that are available under the Handouts tab, and we have 5 there for you to pick up. So please go ahead and check them out. Now please join us in thanking our speaker Ishita for that very insightful presentation and for answering all of your questions. So thank you, Ishita.
Thank you so much, Sonia. Thank you, audience members.
Yes, we hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at XTalks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now. Bye, everyone. Thank you for joining us. Thank you, Ashita. All right, bye.